Some of the things that I want to take back from this lab experience is I can definitely use these techniques in my classroom with the new biotech wing. We've got all the state-of-the-art equipment now, and we can teach these to our students. It's not something that is far beyond their reach. We can actually do it in the classroom, show them the techniques, make them relevant, and one of our biggest goals is to produce 21st century learners. I know everyone's not going to go into the science field, but those that are interested will have an excellent foundation when they leave A.L. Brown High School. Okay, some of the techniques that I can specifically use is something I've been working on actually today is DNA extraction. I realized that's something we've done in the past in our classroom, but I realized today that the technique was not exactly as precise, nearly as precise as it needed to be, and the method that we use, the protocol that we used in here, is something that's very doable in the classroom, and it's going to provide better results, which will mean on the DNA fingerprint or the gel electrophoresis, the student will actually see better results and will have better evidence in their experiment to work with. Another technique is PCR, which actually makes copies of the DNA. We now have thermocyclers in our classrooms, and we can do that technique. The students are all familiar with CSI, and they see them getting the samples of DNA, but what they don't realize is that's not exactly what you need, and it takes a lot of equipment, and one of the things is the thermocycler, PCR, where it makes copies of the DNA so that you have ample amount to work with. Let's see, some of the other things, just aseptic technique, how important it is to have a sterile environment, just learning the equipment, just even learning the glassware. What's the proper piece of glassware to use when you're doing certain things, just to make it, like I said before, just to make it relevant.